Now, let's discuss about embryo culture. So, what is embryo culture? The embryo of different developmental stages formed within the female gametophyte through sexual process can be isolated aseptically from the bulk of material tissue of ovule, seed, or capsule and cultured in vitro under aseptic and controlled physical conditions in glass vials containing nutrient, solid or liquid medium to grow directly into plantlet. Now, what is the principle of embryo culture? The underlying principle of the embryo culture is the aseptic excision of the embryo and its transfer to a suitable nutrient medium of known composition for development under optimum culture conditions. In general, it is relatively easy to obtain pathogen-free embryo since the embryo is lodged in the sterile environment of the ovule or seed or capsule or fruit. So, surface sterilization of the embryos as such is not necessary. Thus, the entire seeds or fruit containing the ovule are surface sterilized, and the embryos are then aseptically separated from the surrounding tissues. Now, let's discuss about the protocol or the procedure of the embryo culture. Capsule in the desired stages of development are surface sterilized for 5 to 10 minutes in 0.1% mercury chloride solution under laminar airflow cabinet. Then wash repeatedly in sterile water to remove all the traces of mercury chloride. Further operations are carried out under a serially designed dissecting microscope at a magnification of about 90x. The capsules are kept in a depression slide containing few drops of liquid medium. The outer wall of the capsule is removed by a cut in the region of the placenta. The halves are pushed apart with forceps to expose the ovules. A small incision of the ovule followed by slight pressure with a blunt needle is enough to free the embryos. The excised embryo are then transferred by micropipette or small spoon-headed spatula to standard 10 centimeter petri dishes containing 25 ml of solidified standard culture medium. Usually 6 to 8 embryos are cultured in a petri dish. The petri dishes are then sealed with cellotape to prevent desiccation of the culture. The culture are then kept or incubated at 25 degrees centigrade and 16-hour light is given. Subculture into fresh medium are made at approximately 4 week of intervals. Now let's discuss about the application of embryo culture. Prevention of embryo abortion. Incompatibility barriers in interspecific and intergeneric hybridization program leading to embryo abortion can be successfully overcome by embryo rescue. In fact, many distant hybrids have been obtained through embryo rescue techniques. Then, overcoming seed dormancy. Seed dormancy is caused by several factors like endogenous inhibitor, embryo immaturity, specific light and temperature requirement, dry storage requirement. So further, in some plants, the natural period of seed dormancy itself is too long. Embryo culture is successfully applied to overcome the seed dormancy and to produce viable seedling in these plant species. Next one is shortening the breeding cycle. Some of the plants in their natural state have long breeding cycle. This is mostly due to the seed dormancy attributed to seed coat or endosperm. The embryos can be excised and cultured in vitro to develop into plant within a short period. For instance, hollies and Christmas decoration plant can be grown in 2 to 3-week time through embryo culture, in contrast to 3 years period required through seed germination. Next one is production of haploid. Embryo culture has been successfully used for the production of haploid, overcoming the seed sterility. Certain plant species produces sterile seeds that do not germinate. For example, early ripening varieties of cherry, apricot, and plum. Seed sterility is mostly associated with incomplete embryo development, which lead to the death of the germinating embryo. Using embryo culture, it is possible to raise seedling from the sterile seed of early ripening fruits. Last application is clonal propagation. Embryos are ideally suited for in vitro clonal propagation. This is due to the fact that embryos are juvenile in nature with high regenerative potential. Further, it is possible to induce organogenesis and somatic embryogenesis from embryonic tissues.